Welcome back to Birds of a Feather Talk Together. Kevin Burns joins us again this week to discuss the Inti Tanager. Kevin is an ornithologist and professor of biology at San Diego State University and goes way back with Shannon and John. Kevin led the research that reclassified what exactly a tanager is, so he's the perfect person to talk to about this newly described species. Kevin tells us all about the story of this bird, from the sighting in the early 2000s in Peru to it finally being described as a species 20 years later. The bird earned the nickname the Kill Bill Tanager due to its bright yellow feathers and black brow, which resembled Thurman's outfit in the movie. We're not quite sure how we feel about the nickname, but hopefully that paints a picture for you. We have a ton of fun with Kevin, so I think you're going to enjoy this one. We also answer a question on a leucistic red-tailed hawk that was seen in Michigan. Okay, go grab your binoculars and let's get into it.
I mean, I can tell the story about how they discovered it, if that's what you...
Let's do that, Kevin. Yeah, sure.
Awesome.
Yeah.
Kevin's the author on the paper, the DNA paper. So both of these.
Well, the paper, we eventually published there's everything in it, not just the DNA, but that's part of the reason they wanted to. Took so long is they wanted to get those DNA samples.
Okay.
But,
So it was first seen like 20 years prior to it being described. Is that right?
Yeah. It was the first seen by Dan Lane and Gary Rosenberg were leading a tour, like a bird watching tour in Peru. And on this road called Manu Road, which is a really kind of popular birding road that goes basically from high elevation up near Cusco down to the Amazon. So a lot of birders like to go on that road and see birds. And yeah, they did one of the stops. You know, that's common. You just stop along the road and try to see stuff. And Dan heard this song that sounded interesting, and it kind of was like a yellowthroat that they thought they might be expecting there. And so he heard it, and then it just suddenly popped up onto a branch and in the open. And so he looked at it with his binoculars and didn't really compute. Then he got in his spotting scope, and then he could see it was something really different. This like canary yellow bird with a pink bill and a black line through the eye, or top over the eye, like a supercilium. And so he got Gary on the bird too. And so Gary saw it, and then Gary got a quick recording, and Dan got a quick sketch, but the bird flew away, like just almost as quickly as it appeared. And so nobody else in the group saw it. And they had this sketch and like a 7-second recording, and that was it.
Was it just an audio recording, Kevin, or was it a video?
No, just an audio recording. So this is 2000, so there weren't, the digital cameras weren't like they are today and things like that. And so on tours like this, you want to record birds, but they're also useful to use playback. So you can record something, and if you bring something in for the group to see, so they always have a recorder with them on tours like this.
These are some of the best bird watchers in the world. This isn't like me seeing something that I don't know, which would be virtually everything. I mean, if they say they see something and they're confused about what it is or don't recognize what it is, then there's something for real happening.
It's a big deal.
So then you pay a lot of attention when people like that are saying, I don't know what this is.
Yeah, so they know all the birds there, right? So there's something weird, but they didn't see it very long, right? So it's frustrating. And I don't think they told the rest of the group about it even. And that's probably good, because you never, when you're at a bird trip, you never want to hear what everybody else saw that you didn't, or someone else saw that you didn't.
Well,
Yes.
well, it's even worse when you don't even know what to call it, and you're one of the experts.
Yeah, exactly.
Imagine how angry those people probably were on the trip who weren't told about this bird.
Oh,
Hopefully they were happy with all the hundreds of other birds they saw. And, 20 years later, maybe they learned that this bird was on their trip, but, what do you do? And so then they went back the next year, and same spot, didn't see it. And then back the next year, same spot, didn't see it. And then I guess Dan was sort of kind of doubting that he ever, really, what did they actually see? I saw his sketches. Sketches really good, and he's an artist, so he started thinking about maybe it was some kind of plumage aberration, or maybe the song was like a Vireo song, or I don't know, because those sort of memory is kind of a weird thing, right? Human memory. But anyway, in 2003, they went back, and as they pulled into the same spot, they heard the song. And this time they got a decent recording, played the recording, and the bird popped up for a really good view. And so they all saw it, and they had it in the spotting scope, and nobody had a film camera, but I guess Gary was telling me this. I just saw him last week. He had a digital camera with him. And so he was digiscoping things like we do now, where you put the phone up to the scope or something, but with a digital camera.
okay.
Because, phones weren't where they were either in 2003. But all these other people on the trip, right, paying to see the birds. So they got to go look through the scope first. Like one by one, everyone's getting a look at the bird. And then finally, Gary goes up and takes this picture. And all he gets is a branch because the bird flew off.
Oh, no.
So they had no photograph or anything, but they had much better recording now and much better feeling of confidence that this was something new and different. And, but that's not enough to describe any species, right? You need hard evidence, tangible evidence. Even a photograph probably isn't enough, so that's not enough to really get them where they want to be able to describe it in a paper. That's why it took so long, because it took a while to find a lot of these birds. And I guess the breakthrough maybe was in 2011 when a different researcher, who I actually don't know, Frank Wright, was working in Bolivia and found a large population of these birds breeding. So together and breeding in one place. And so that was in 2011. And so once that happened, they were able to launch additional expeditions to study the birds in, I think, 2012 and 2018 and 2019. And that's when they got samples that get sent to me for DNA analysis and things like that. So that's part of the reason why it took so long. And they just wanted to have everything too, right? All the biology, all the natural history that they could in the description. Of course, the DNA evidence to help us tell, what the heck this bird even is, because when they first saw it, the first thing that came to mind was this family of African orioles, which they're not related to our orioles, but they're superficially similar. Some there that are bright yellow with the black stripe through the eye. But that's fairly improbable, right? So then they started thinking about things like tanagers, because as we talked about on an earlier podcast, the tanagers were kind of a big wastebasket. There's so much variation in them. It's like, well, maybe it's part of that big bin of variation, and maybe that's where it went.
And then was this discovery happening in the middle of all the reclassification of all the tanagers? Was this all kind of at the same time?
Yeah,
I'm sure that made it a lot more difficult.
Yeah, but I think because I was sorting through all that mess and figuring out piece by piece, I think that was the reason why, I was the one to send it to it. I'm grateful that they did. But yeah, around 2000, I had some early papers with early 2000s with some with Shannon, where we kind of pieced together some of it. But 2014 is when the biggest sort of paper came out where we sorted everything out. So that's right when they were doing all this stuff. So, and we kind of did the analysis separate on the side so that, it wasn't going to be put into the paper, the results weren't going to be buried in with all the other tanagers, because it was going to be part of the species description. So we couldn't necessarily include it until the species description was done. So,
But I doubt it would have gotten. I don't know. Do you think it would have gotten its own genus if you didn't have the molecular data to show how genetically distinctive this bird was?
That's a good question, because there are distinct things about it, but definitely Dan wanted to wait until we had the DNA, and he felt the DNA really showed that evidence. So there's other things distinctive about it. The pink bill, the bright yellow color. It sings like during the day. Its song is very different, even from the birds it's related to. And also females and males apparently both sing, which is not that common of a trait among birds. So we got the DNA and we figured out, yes, it's a tanager, just like they thought. And then we did sort of two sets of analyses to try to figure out what kind of tanager it was. When we first got the samples, I had a student named Luke Klicka, I'm going to give him a little shout out, who did a lot of the work on the initial steps where we had like a handful of genes, like 5 or 6 genes. And we were able to figure out it went into this one clade of tanagers, which we call the ornamented tanagers, because they have a lot of birds with colorful ornaments, like the birds in Ramphochelys, the silverbeaks, which Shannon's worked on, these bright, shiny bills. And then it has a lot of birds with crests. And guess who else has a crest? The Inti Tanager. So it fit right in there. But we couldn't tell if it was closer to, there were 2 different ones, the black goggled tanager and, oh gosh, I'm blanking for a minute, the gray hooded, Eucomedes is the genus, whether it was one of those 2. And both of those have their own genus. So Shannon, I'm circling back to your question about, why is it in its own genus? Because both of those were in their own genus. And so the fact that we couldn't sort between them because the branches connecting them were so hard to tease apart, at least with that initial data set, was some evidence that it should get its own genus. And then we finally, in 2019, another student of mine, between 2014 and 2019, technology got a lot easier and cheaper, and we were moving to do different things in the lab. So we were able to actually get like 5,000 genes. These are ultra conserved elements, is what they're called. And to look at this question, there we were able to pinpoint that its closest relative was this black goggled tanager, Trichothropus. And so the fact that the branch between all 3 of those is pretty short indicates it probably deserves its own genus.
Well, what would have happened if all of the branches had been short, like they were all pretty closely related? You probably would have sunk one of the genera, and there would have been 3 fairly closely related species that aren't that genomically different from each other in one genus.
Oh, yeah. I mean, I guess you could do that, put them all in one genus. I mean, you could do that, I suppose. But again, this, I guess it highlights sort of the fact that these taxonomic categories aren't real. They don't represent anything really biological. They're just kind of places that different taxonomists have drawn circles around things and said, okay, let's describe this as a genus and this is a family and this is a so forth. But, there's some effort to try to get those matched to certain dates and timelines and so forth, but it's kind of hard to do. Mainly it's kind of just based on kind of degree of morphological divergence, and birds are probably split into more genera than they need to be compared to other taxa, but there's pluses and minuses going both routes. But I don't know, it's a pretty different bird than the other ones. That bright pink bill, can't think of any other tanager with a pink bill. So I was happy with the decision.
I was just going to say, until you get DNA data, can you really describe a bird as a new species these days? Or is that kind of like necessary at this point?
Unfortunately, I would actually say it's not necessarily necessary. And the other thing is there have been species that have been described based on when they've had DNA data and there are no DNA data differences. There are species that have been split when people know they hybridize. So the idea of what a species is something we could spend a lot of time on. In this particular case, one of the really spectacular things is just how different this bird is to its sisters.
Yeah, I think most new bird species descriptions these days, it's like something that already has something it's really closely related to or very similar to, or a lot of times the discoveries are made in the lab with genetic data or in the museum by a careful study of differences among existing samples. This is just something like completely new. But, not necessarily for birds, but for lots of organisms, we still are just, there's lots of species described all the time. We don't even know how many species are out there, right? The estimate is there's like 8 or 9 million species, or maybe some estimates up to 100 million, but we only have about 2 million species described.
Oh,
So
wow.
it's kind of crazy because each of these species has an important story to tell and things that we can learn from them in terms of adaptation or even useful chemicals and compounds. And then to keep ecosystems functioning, right, we need to know all the players. We need to know all the species. And, if we don't know that, we don't know how they all interact. And a good analogy here, and this is, I think, from David Hillis at University of Texas, is like conservation biologists are like car mechanics that are trying to keep a car running, but they don't know what all the parts are. and the parts are falling off before they even know what they do. And so, there's so much biodiversity, and there's too much undescribed biodiversity and not enough biodiversity scientists out there to sort of get us to where we need to be.
And even in a group like birds, which people would say is among the best known taxonomically, I like to point out to people that there's a publication that was done by a whole bunch of ornithologists for the American Ornithological Union back in the 70s, and they said 9,000 species of birds, and we basically described them all. And if you go look at birds of the world today, we're at 11,400. So, and there's still new things being described all the time. And so what I think some of that is just literally there's splitting up of diversity in ways that with new, with molecules and things across geography that makes things, people think they're different enough to count as a different species. But then there are birds like this Inti Tanager, which are just completely different. And, the neat thing about it from detectability is that the area that Kevin was talking about in Peru is actually their wintering grounds. And so they're only there part of the year as far as what they know about them so far. And while that's really well traveled, the area to the south of it where the birds breed in northern Bolivia is super isolated, and nobody goes there to speak of.
Kevin, how many tanagers migrate?
Not very many. I don't have a number off the top of my head, but that was one of the big surprises. I mean, I would guess maybe 20 or something. There are not a whole lot of them that are in the temperate zone of South America. But what's weird about this bird, and that was one of the big surprises, it's an intratropical migrant, so migrating within the tropics between this spot in Peru and then hundreds of miles away in Bolivia. And so that place in Bolivia, like John said, isn't as well studied. People had been there before to study birds, but in the dry season, when it was the birds weren't there, because the birds were up in Peru. But then in the wet season, when they're there, it's really hard to get to. I understand it's like a 22-hour bus ride from the nearest place you can get a bus, then a hike through all of these streams and rivers, which may or may not be able to be crossed to get to the spot where these birds are.
Oh, wow.
Yeah, I obviously had not been there. I've been on the road and where the tanager was seen in the dry season and tried to see it there with no luck. But that explained kind of the mystery why they didn't see big populations of it at that one location.
So, and Kevin, did the birds look the same in Peru and Bolivia, like based on whether they're breeding or not breeding? Like, would they look pretty similarly?
Yeah. As far as I can tell from looking at the specimens I've looked at in photographs, they seem the same. I don't think they don't change colors like some of our migrant birds do here.
And this is kind of a follow-up to RJ's question about the DNA. We've wondered, I guess, first off, is there a way to get DNA from a bird without killing it?
There is.
Yeah.
But the thing that happens is that without the representation of the specimen, you can't go back to the original source. And if you've spent any time in museums, you see this all the time, which is you start studying something and all of a sudden the thing you studied isn't either what you thought it was. It's composed of a whole bunch of things that you didn't know about. Some of those things are not all each other's closest relatives, and they go somewhere else in the tree of life. And without the actual physical specimen, you have no way of tracing all these differences back to the individual. So you're left with very uncertain. Did you get an aberrant individual? Did you get a hybrid? Are there more like this or not? Because sometimes they really do look the same. You can come into our collection and you can open a drawer and see all of these blackbirds, for example, all laid out together. Someone studies them for real, in this case, using DNA. Then we realize that these birds had a physical characteristics, eye color, that documented that they were different, and not that it was maybe an immature bird or a juvenile or something like that had a different eye color. Well, you don't know. And now we do know a little bit, which is that these things with different eye colors, not only aren't they in the same species, they're nowhere near each other's closest relatives. And one of the things belongs somewhere else, has a different elevational range. So, and might be one of the rarest birds in Africa. Uh, all looking exactly the same in a drawer. You'd never be able to tell them apart. The labels and the fact that people pay attention to detail can give you some clue. Sometimes you don't know until they sing, and all of a sudden they sing really differently. You can't tell that from a picture. You're not going to get that out of a DNA sample, a blood sample only. So yeah, people can take blood samples, and a lot of population analyses require you to do that. You can't sacrifice as many birds as you would need, but to describe something, you can't, in my opinion, this is variable. You can't just use a picture. You shouldn't just use a DNA sample, or a piece of blood or a feather or something like that. You need to have what John would call the extended specimen. You need to have the geographic coordinates of where it was found. You need to have the organism so you can go back and actually look at it. Uh, Eventually, you want to have males and females, and you want to have things from across its geographic range and things from throughout the life cycle. So you want juveniles all the way up through adults. And most things, we don't, even something as common as American robin, we do not have enough samples to study those kinds of questions across one of the most common birds in North America. So you need to have samples. They need to be in museums. They need to be cataloged and available to other people to study. So I'll get off my soapbox now.
Well, here's an interesting example with that Inti Tanager. So, Kevin, there was a specimen collected in Peru as well, right? And did you guys have genetic data for that you were able to look at?
I don't think so. I think all that we were given was stuff from, I'm trying to remember. I can look at paper, but I think the stuff we had was all from the breeding location.
Okay. So right now, Amanda, so the data that they have, it suggests that there's migration, but maybe there are two populations. And if you had if you had genetic data, you might find morphological data if you had a specimen to say that there are two populations of this thing rather than one. And so there are various questions that you can answer with specimens that correspond with the genetic stuff in ways that are really useful.
Yeah, think of the giant hummingbird that we did a podcast on a while ago. I mean, if those things weren't represented in collections, there's no way that Jesse Williamson would have figured out even the beginnings of such a complicated, weird, strange story. Because when things don't fit your preconceived patterns, your preconceived notions, and everybody has preconceived ideas, some pretty well informed based on the expertise and knowledge of a person, that doesn't mean that everything turns out the way you think it might. There are surprises hidden everywhere, and every collection is full of dozens of surprises, if not thousands of surprises. That's why they're so valuable. And then our technology changes, and we can ask different questions and get different answers associated with things like what makes those birds yellow, what kind of pigments go into the black, and are those pigments in black or yellow the same as other tanager pigments, or are they different? Is there ultraviolet coloration? If you don't have a specimen, you're not going to know that because our eyes are inferior to bird eyes.
I'll just add one more thing and it's along the same lines. Like in my work with tanagers, and there were at least two samples, I can't remember which species they were, where they were coming out in a weird place in the tree. And so I wrote back to the museums and say, hey, is this tissue right or whatever? And then I got a response back, oh yeah, I misidentified in the field as X species. But now the tag says this. We realized it was this once we got it back to the museum. And so then it made sense. But, if it had just been a blood sample taken in the field, I probably would have published something incorrect about that species because it was misidentified in the field. And that's no critique of the field biologists because they don't have the museum in front of us and they may or may not have a field guide that's decent. They're just trying to identify, what's in a new place that maybe no one's ever been before. And so, you really need those museums to sort of really clarify the identification.
And this is why public databases that you deposit information is, whether that's our collections being online or the DNA sequences going into something that NIH manages called GenBank, are so important, because that mistake that Kevin was talking about, if he had published it, the only way he could get that paper published is to put his DNA sequences in this public database called GenBank. And then every person who uses that sequence afterwards has got it incorrect, right? So it
Wrong.
perpetuates a whole series of wrongness through time. But GenBank, when they force the people to put the actual collections numbers in, you can then update the identification that's on that DNA sequence and hopefully stop the chain of terrible science that could be done and inappropriate hypotheses that get generated from it. But this is a serious problem. GenBank is full of wrong things because maybe there was only a blood sample or a feather and there was an incorrect identification. Maybe you were lazy and you didn't put the museum information in, or you didn't realize you'd made a mistake and then you didn't go back and correct it. So there's lots of things like that we have to be really careful and guard about. And then every once in a while, they were right. There is something weird. It doesn't go anywhere near where anybody thought it would. We've all had that experience too. You're like, I don't believe this. This is not right. And then you do it again and again and again with more and more specimens, and it still does that. And so you're faced with the data blowing your preconceived ideas out of the water and making you reconsider what you thought. And that's good for scientists to be taken out of their comfort zone, put somewhere new.
I think in this particular example, it really highlights the other thing, which is that you bring people together with a lot of expertise on these projects these days. And I love that aspect of it. You know, you got people doing the molecular work, you've got people doing the field work. There are Bolivians and Peruvians involved in the project. I mean, it's just, you know, things have changed a lot from when people were first going out and collecting specimens in various parts of the world back in the early 1900s.
You know, and you can't use a spectrophotometer on a photo and figure out that it actually has different underlying color patterns. You can't measure as easily a photo. So, you know, nothing. And besides, when you look at birds close up, they tell you all kinds of stories that a picture, which it's okay, but a picture can't do.
I want to make sure, I just want to revisit that word discovery here and make sure that that we talk about that, because it's incorrect to say we discovered this bird, because we don't know what past peoples may have known about that bird, or even though the area is sparsely populated now, I think that if anyone else had seen the bird before scientists. So we really just described it to science. We didn't discover this bird. So I wanted to clarify that just in case that wasn't clear to people.
I was just wondering if there's any kind of like influx of birders or, you know, people that are wanting to go see this after something is described. You know, are people all of a sudden flocking to the area trying to get a glimpse?
I don't think there's flocks of people, but there definitely are people going there. I see more eBird records now, and I've seen at least one tour leader going there and things like that. So, but I think the area is really hard to get to. So I'm not sure it's going to be like, you know, on the birding circuit necessarily. But that, I think, to me personally, that's a little bit of a concern, because you don't want to have too much impact on the habitat. So I hope that on the ground people are being cognizant of that.
Sure.
The area that it's in is adjacent to a national park and it's protected. And so that's why I think in our paper we wrote data deficient in terms of conservation concern, but we really don't think it's of conservation concern. There's I guess a road nearby that's being maybe developed that could change that. But right now it seems like it's doing pretty well, all things said and done.
And I wanted to know, if it weren't for because it sounds like in Peru, they kind of saw a very limited number, or they saw one or two or whatever. And then in Bolivia, that's where they were breeding and there was a greater number. Is that correct?
Yeah.
Yeah. And then, so is RJ and we've talked a lot about like we're interested in like the kind of some of the nitty gritty stuff you guys do as far as like taking specimens. Was it not until the breeding population was found in Bolivia that people were like, okay, now it's safe to take a specimen?
Yeah. And that's an interesting issue that people are thinking about. You know, if you're doing survey work and you get something that's completely new, you're going to wonder how many there are out there. And so
Right.
there's a very classic, recent, within the last 20, 25 years, case of a bird that was found in India that's been accepted by everybody as a new species, where the guy the Indian researcher that found these things was pretty convinced there were no more than five territories, I think. And so he didn't collect a specimen, but he put together a bunch of information about the bird, and the scientific community basically accepted it. I mean, one of the things he said in the publications was he would take a specimen if it turned out that there were more individuals out there that they knew about. And, personally, I don't have any problem with that. Yeah, I think in this particular case, they knew from the research that had been done by Frank Wright that there was a population in this area, and they wanted to make sure that they got information, including a number of individuals, to be able to look at the kinds of things that we look at with specimens on a regular basis.
And you need permission to do these things.
Yeah.
You need permits. So you can see something new, but the first thing you do is you go back to the permitting agencies and say, we found this here. Will you give us permission to sample it? And then there's a discussion of how many there are and what the efficacy is of doing that. And this is not something, you don't just run out and grab it new. You might run out and see if you can grab poop or something like that, a feather, any part of it, just so that you do have some DNA ability to trace something. And even that needs permits. You can't export birds from these countries without permits. You cannot import them into the US without proper permission. So it's not rogue ornithologists running around, doing that. It's not the 1850s anymore, or even 1950s anymore. Almost all of these projects are done in collaboration with scientists in the country and in the regions that these birds are coming from, because the only way to protect them is to generate a cadre of scientists who are in these places, who will make sure that governmental laws allow this, that even, that land is protected. I think those things are really important these days.
And just to get even deeper, we watched last night, we watched the movie Silence of the Lambs.
Oh,
And if you watch that movie,
Halloween.
you would think in the spirit of Halloween, we were watching it. And but if you watch that movie, you would think if you were training to be in the FBI, like 90% of your time is on the shooting range or like working out. That's what that movie kind of makes it seem like. And I was wondering, like, when you guys do decide that it's appropriate and you have permission to collect a sample, is it the actual ornithologist, like, who does the shooting, or do you hire like a professional to do it?
The actual ornithologist. But then past there's been like professional collectors, like, decades ago, and that's all they did, and then send stuff to ornithologists. But these days, I think the ornithologists are the ones doing it, usually.
and
Yeah.
training people who come after them to do it too. Right. Hopefully with hearing protection these days, but
Yeah. So like Is it like Silence of the Lambs? You guys spend a lot of time at a gun range?
See,
I'm kidding.
No.
see, Amanda,
I'm joking.
Amanda,
John, what were you going to say?
I was going to take that a completely different direction and say, no, we're more like the guys in the insect collection who
Yeah.
ask if you like beer.
Those were, oh my
We
God.
were laughing.
John, I have to tell you, RJ literally said, that's John and Shannon.
Yeah, they had their specimen collection. We were saying they're like Maureen. We were like, oh, that's so
Yes.
cool. We know like a lot
We
more
know
about this now.
The field museum person like that.
Yeah,
Yes. Maureen seemed a lot cooler than those guys.
Bring it in. We'll tell you what it is. And yep.
Maybe that door is too
Yeah,
far flung
like
open
beer.
for what we're really like in real life. That they could have made us cooler.
Well, I was going to ask
That's all.
Kevin what you think about the Kill Bill nickname for while we're on the topic of movies.
Well,
Is that a good thing or a bad thing, or any thoughts on it?
I don't particularly like it. Now I have to say I haven't actually seen the movie. But,
Oh, no.
the colors are right. You know, the yellow of the bird and the yellow of, you know, the actress's jumpsuit and the black line across the eye and the black line on the jumpsuit. I guess it makes sense. I don't think Dan likes it either. It wasn't. I forgot who came up with it, but I think it was some other bird or something. Because you know the bird didn't have a name, but people knew about the bird, so it didn't have a name, and so people were just kind of floating things around, somewhat tongue in cheek, or I guess to do. But that one kind of stuck. So. Yeah, I don't think it gives the bird justice. I'll say I like the name we gave it, Inti Tanager, which Inti is a Quechua or Maya word for sun,
Okay.
which kind of trying to honor traditional languages in the region. And so sun because it's colored like the sun, and it actually apparently sits up in the open and sings in the sun in the middle of the day, which a lot of birds don't do. They mostly sing in the morning, things like that. So I like that, sun or Inti Tanager. Same with the genus name, Heliothropus. Translates to sun tanager.
Very
And
cool.
we should talk about the O'Neillii part too.
Yeah, so it was named for John O'Neill, who actually was on that trip to Costa Rica with Shannon and John and I. He's a famous ornithologist from LSU who's described many species in his own right. And so he worked in Peru for years and supported a lot of Peruvian ornithologists. And so Dan, that was Dan. You know, this is really Dan's bird, so he's really the one picking the name. And so was Dan wanted to honor John with that name.
Oh wow.
But I also want to point out here that, you know, something going back to the very beginning of this discussion, which is that, you know, bird tours are going around the world on a daily basis into various areas. And Dan and Gary were leading a bird tour when they first saw this thing. And so, you know, again, there's oftentimes those bird tour leaders have ornithological backgrounds and in many ways are practicing ornithologists, but you know, they have a good handle on what's in the area. And so when something weird shows up, they notice it.
And they document it in eBird. So there are public databases now that let you understand the distributions, maybe even population changes, size changes through time. So these are really important ways of preserving birds is for people to know where they are and for people to look at them and count them. Get enough information about them that we can know what their population size might be like, what their endangered status might be like, and all kinds of things. How many eggs does it have? What does a nest look like? How long do the babies stay around the parents? How far do birds go from where they're born to where they breed? Which the rented episode is really important from that perspective. So what's its diet? That's another thing the specimen can help you do too, which is to sequence the gut contents, because sometimes we don't know what these things eat until we look in detail at what's inside their stomachs. We don't know what their microbiomes are. We don't know what their blood parasites are. All of these things come from having the actual specimen What kind of feather lice does it have? What kind of mites does it have? What kind of flies are on it? I know it's gross. Does it have worms inside? And then how do those compare across the things that are found in the same habitats or across tanagers? These are things we can only know if we actually have samples, which is why these days we do so much work in the field when a specimen is sacrificed so that we can maximize the information we can get from it. So that bird goes from Peru to Bolivia. What happens to its microbiome in these two places? We know migratory birds in North America, there are differences that can happen along that route. What happens there? We don't know. Does it eat different things? We don't know. So it really does take a lot of people with a lot of expertise to start documenting these things and to give us enough information that we can truly understand the bird from a holistic perspective. I don't want birds to die, but if they do die, they need to be in museums where they can be studied for forever. Each individual has its own story, just like each individual person does. But they also tell the story of the species they're nested in too.
And we've been talking about, Kevin's phylogenetic work over the years, looking at the relationships of these tanagers and rearranging them in a way that we get much more correct based on DNA data. That's, I think the inferences are real strong. And I'm always fascinated when something like this shows up is about the biogeography of it. How did that evolve to be limited to this particular area in northern Bolivia and southern Peru? And what do its relatives tell us about that relationship with respect to the history of that part of South America?
That is so super odd, that kind of migration pattern. I mean, do you think that thing was more commonly distributed and it's now in relictual populations?
So there is another species of tanager that has a distribution like that and behaves like that, and it's called cone-billed tanager. It lives in dry forests in northern Peru. I don't think it's associated with bamboo like differences of are for Inti Tanager, but it migrates out into actually, now that I think of it, it actually migrates down into southeastern Peru. And so some of that I think holds some really spectacular links to the paleo history of that part of the base of the Andes. And these dry forests must have existed long enough for some of these species to have evolved in them. And then climates changed and you had some forests moving around and responding to that by doing what they do now. And so, those are all questions that, man, it's going to be a lot of fun for some to figure out.
Kevin, you've done some really fun things in your career.
I've been very fortunate and lucky to be able to do some of this. This project in particular was unexpected, and just very thankful to have been included in doing that and solving that. And it was fun to work on because, we'd have up on the whiteboard this big word mystery bird or mystery tanager, and everyone would kind of guess what was going to happen. And then, you get the DNA and then you see where it's at. And then, it took several years for that to happen. So I don't know, it was fun.
That's awesome. I mean, that's scientists. Being a scientist, we're super lucky because we get to learn things all the time that, and that's our job, is to learn things. But every once in a while, or maybe pretty frequently actually, you learn things that nobody knew before. Not just that you didn't know, but that nobody knew. And that's like the tiny little kernel that is at I think the center of every scientist, no matter how jaded and famous they can be. It's still that idea of discovery and how driven you are to figure out the way the world works. Why is this? What is it? Why is it this way? What does it do? Why does it do it? It's so exciting and nerdy. I know I just sounded super nerdy when I said that, but
I think you sounded cool, Shannon.
I think you sounded cool too.
Yeah, when you said that, I was thinking some of the things I get really excited about because I found out very few people would really care. It's the dirty part.
Oh, well, I make John care about them all the time. Hey, did you know this? Hey, did you know that?
That's awesome.
But this was one people did care about. It got some attention, which was fun.
That's cool.
Kevin, I think you should watch Kill Bill.
Oh,
I just may have to, I thought I watched everything during lockdown, but I guess there was one movie I missed. So maybe I need to watch that one.
I thought it was funny. I saw Kill Bill came out in like 2003, so it must have been right around the time. It just happened to be like the movie with a yellow, if there was another movie that came out with a yellow character, maybe would that be the nickname?
Yes.
Yeah, exactly. I think that was the movie at the time. Maybe if it was in the 70s, it would have been called the Woodstock Bird or something.
Yeah, yeah.
Snoopy's friend. Yeah.
Yeah, John, I was wondering what movies were out when the Willard's Sooty Boo Boo was described. Do you?
Not much. Yeah,
Not
I'm
much?
trying to think. Yeah, I've never been inspired sitting in a movie theater,
Okay.
except when the guy identifies what kind of caterpillar larvae you're talking about in
Yeah.
Silence of the Lambs.
Let's lamb.
That movie's terrifying.
It was.
Oh my God.
It was so horrifying. We were like, we had
It
to pause it partway through.
Yeah,
Yeah.
it puts the lotion in the basket, which has become
I
it's become a thing in our
I-
DNA lab. It's like yeah. Oof.
remember I saw that movie with you, Shannon, and I went back to the lab that night, wrote my master's thesis. I couldn't go to sleep. I just had to work. I could not sleep.
That's awesome.
Just
That is awesome.
absolutely terrifying. Oh,
That
I
is
never
so
think
good.
of Ohio or a sewing machine in the same way ever
Oh.
again.
All right. Well, I think we're kind of getting close on time. Is there anything else on the Inti Tanager we want to talk about?
Hard.
We've been pretty good.
If it's not an easy bird to get to see, but it'd be worth the trek to look for it. It's a neat part of the world.
But scientists have put it into the public domain by taking all the knowledge that we have and putting it out there. So it's the beginning of that bird's, moving into the future. So take the past now this way. The future will tell us more.
Well, we do have that mailbag question about that red-tailed hawk. It's our mailbag question. This is from Kelsey. She's from Michigan, and she's writing. She sent a couple pictures and said, "Have you guys ever encountered and I'm sorry, I might botch this word, a leucistic bird? Am I saying that right?
Leucistic.
Leucistic?
Yeah.
Okay, sorry. You guys ever encountered a leucistic bird? I had recently been keeping an eye on a leucistic red-tailed hawk in my area, St. Clair County, Michigan, and want to know who to contact and if I should contact anybody about getting her banded and data collected. She is a full-grown adult and has been doing an amazing job at surviving with her strange coloration." And she sent a picture, and it's not fully leucistic, but there's a lot of lack of pigment on it. So there's a little bit of brown and then a lot of white on the picture. We can post it on our social.
Yep, the bird has a very red tail, which I think is really interesting. So it's a typical red tail from that perspective, and then almost no other. And we were talking about this. I mean, one of the great things is we talked to Bryce Robinson at Cornell University, who's working on his PhD on genetic structure across the distribution of red-tailed hawks, and he immediately wrote back and said, make sure you put that those photos up on eBird, because then they go into a database where people can find them. And this whole issue with sort of these birds that have partially white plumages leads to all kinds of different interesting discussions about how that happens. And there's probably a genetic component, but then there could be some other components associated with things happening as feathers are growing. Certain things get shut down somehow, whether it's, I don't know, disease related or something else, where maybe that's the source of some of these birds having partially white feathers across their body, but not entirely white. And it's not an albino because it's not completely lacking pigment. And so, these are interesting things where they clearly happen out there in nature, and we don't know much about it. At one point we should talk about one of my favorite birds that was described as a new species recently within the last 20 years or so, called Araripe manakin, which is a bird that is a small isolated population of a much more widespread thing. And the big difference is the males have a white body, basically. And so there can be implications for the speciation for speciation if there's a possibility that it gets established in the population. I don't think that's going to happen with these red-tailed hawks, but they
White
clearly
feathers,
happen.
having white feathers like that, or a little bit of white body parts, is not that uncommon. And we've had, especially when we were spending so much time at home, and I got to know virtually every house sparrow that was in our yard. There's more than one that had partial white feathers, and so we would chart it. Is it here? Is our friend here? Um, and so if you spend enough time looking at things within a species, you're likely to find an aberrant individual, including some really bizarre things that are bizarrer than white, which is you can have birds that are half male and half female plumaged because of some developmental thing that happened early on when feathers are being laid down. So you can have all kinds of really bizarre things that go on, whether the environment, pollutants, parasites, DNA mutations while the bird is developing. All kinds of things can do that. If you're completely albino, you probably don't last that long. You don't have pigment to protect you from getting a lot of ultraviolet damage. Your eyes are really vulnerable because they don't have pigment in them. We have pigment in our eyes for a reason. So, and you're bright white, which means to every predator, hey, come look at me. So the chance of you making it out of being a baby bird are pretty small, probably. So you just don't see that very often. But white patches on birds are not uncommon. So if you spend enough time looking at a species of birds, you'll find some that have. And red-tailed hawks are big. They sit out in the open. So they're the most likely kinds of birds you'll run across to see these white feathers. But it's really cool. And she reached out, and now that observation is in a database. And if Bryce wanted to, he could get permission to go out and capture that bird and take some more photos, take a blood sample from it, take some feathers from it so we can understand why it looks that way.
And red-tailed hawks are interesting because across their geography, they actually have some birds some melanistic phases of plumages are really common in birds of prey. And one of the things he's been studying is actually this bird that breeds up in central Alaska, a population of red-tails called Harlan's hawk, which is basically a dark version of red-tailed hawks. And most of the population seems to be consistently dark like that. And so there are differences that can mean something associated with the geography of birds sometimes.
Sure. If those changes in plumage happen in an egg and a sperm, then it's going to go into the next generation. Most of these don't do that. It's going to be just the individual that has these bizarre, weird feathers. But the mutations that happen in the germ line in your eggs and sperm, those are the things that matter from an evolutionary perspective. So, that's likely one of the ways in which we do get new species is there are these kinds of mutations. Then they do get passed on, and then they can drift away from other populations, or they can be selected on, or whatever. But and maybe some populations of bird are just more prone to making mistakes or having errors happen when they're making feathers. I don't know. That's just a rampant guess and speculation.
Are birds that are actually white, do they just have a lack of pigment, or do they actually have the color white in their feathers? Like, is there a difference between?
No, there's no color white, but some of them have underlying structural color. So you think it's white, and it might look white to your humans' inferior eyes, but it can be full of color and pattern to birds who can see things underneath. And when you have a spectrophotometer, you can actually measure what is happening in other wavelengths that we can't see. So you can't make the assumption that there's actually nothing happening in a white feather, because there can be things that happen underneath. So it is a lack of color, a lack of pigment that produces the white.
Okay.
But RJ, we were up in on the Wisconsin border yesterday, and we saw some trumpeter swans, and I was just thinking, like, how do swans get white, and what is that white? And
Yeah.
it's basically something that works for swans.
Yeah.
But geese have it too. So there's lots of white in that family of birds. Interesting.
Well, Kelsey, thank you for writing in and for contributing. We hope we guided you in to submit that just so we can research it. All right, I think that's it on this one. Does anyone want to close it out?
Yeah, I'll close it out and just say that when you go to Birds of the World to look up Inti Tanager, and you should do that, don't be distracted by the fact that they decided to put the female, which just happens to be less brightly colored, as the tag for the bird when you first get into the species account. I don't know why they did that.
Okay.
Well, it's half of the species are female. Why not?
That's a little biased, John.
It is.
Oh my goodness. I cannot believe you said that out loud. Holy cow.
Wait, John's just saying because the male looks cooler, right?
Who's to say
Right?
that makes it cooler? The interesting
Well,
things that
he's
happen
brighter.
in tanagers have to do with the way female plumages change. Oh my God, John Bates.
Fair enough. Fair enough.
John, you're in trouble, man.
Wow.
I know.
You're going to want to edit that out.
No, I don't think you should.
I can't believe you did that. That is a way to end
Right,
it,
that's-
John, but not the best way to end it.
It's a good place.
Wow. Thanks everyone for joining us this week. We appreciate all of you. If you have a question for us, you can reach us at podcast.birdsofafeather@gmail.com. All right, we love you all. See everyone next week.